This video covers the theoretical explanation about the determination of fluoride content by colorimetric method. The fluoride which present in water can be determined by spectrophotometric or a colorimetric method. So the basic principle involved in colorimetric method is the reaction here, particularly in the fluoride, it's a reaction between fluoride and a complex zirconium with an organic compound called SPDNS, SPANS. The full form that is expanded is sodium 2-para sulfophenylazo 1,8 dihydroxy 3,6 naphthalene disulfonate. Usually the fluoride, when it is combined with the zirconium SPANS reagent, a complex, the fluoride react with this complex and dissociate this portion of the complex into a colorless hexafluorozirconia and reagent. We can express this way, like I'll take a zirconia SPADNS reagent. When it is combined with the fluoride ion, it breaks the complex into zirconium hexafluorozirconia and SPADNS. And this is a pink colored complex. So this complex is a pink in color. As the quantity of fluoride ion increases, the color developed by the complex decreases. Okay. So it dissociates to forming a hexafluorozirconia and the reagent. As the amount of fluoride ion increases, the color developed by zirconia SPANS complex decreases. A calibration curve is in all colorimetric estimation. A standard calibration curve is plotted absorbance at 570 nanometer for a series of standard solution versus concentration of fluoride or the volume of the fluoride ion solution taken. By measuring the absorbance of the sample after treating with a reagent complex, the amount of fluoride present in the sample can be determined. So this is an organic compound called SPADS. So then, while estimating the fluoride content which present in the water sample, it is necessary to separate the fluoride from the sample by distillation, which otherwise leads to the error due to interfering ions. The procedure in the estimation of fluoride content in a water is divided into three. The one is the preparation of the reagent, the second step is the calibration curve, and the third step is the determination of fluoride contamination in water. In the preparation of reagent, to prepare zirconium SPANS reagent will take around 950 milligram of SPANS reagent. So we'll take a 950 milligram of SPANS reagent in a 500 ml of reagent water. Okay, in a 500 ml reagent water, 950 milligram of SPADNS is taken. To that, we'll introduce a little amount of distilled water until the crystal should dissolve it completely. Then it is diluted to 500 ml. Then we'll take the other compound, that is an acid reagent, that is taken in a round bottom flask. That is again here we'll take around just pictorially I'll represent this one just to memorize. Here we'll take 133 milligram of zirconyl chloride octahydride. Okay, to that 25 ml of water is added. The remaining 350 ml of hydrochloric acid is added, particularly concentrated. Then it is diluted to 500 ml. Then equal volume of these two solutions are mixed to get a zirconyl SPANS reagent, that is zirconyl acid reagent. This is a preparation of zirconyl SPANS reagents. Okay. The second step involved in this is the preparation of calibration curve. For that, we'll take 0.221 gram, just a 0.221 gram of anhydrous sodium fluoride that is dissolved and made into 1 liter. That is 0.221 gram per liter. For per liter, we'll take. Then this solution is diluted to made into 10 milligram per liter of fluoride. Okay, then again it is diluted. Then we'll pipette out 1, 2, 3, 4, 5, 8 ml of stock solution into a 50 ml of standard flask. We'll take 8 standard flask like this. 8 standard flask we'll take. To each standard flask, so we'll pipette out the same quantity of, each standard flask, we'll pipette out the increasing order of like 1 ml, 2 ml, 3 ml, like so. In this here we'll take a 8 ml of the stock solution. To each standard flask, 10 ml of reagent is added. Then to each standard flask, 10 ml reagent is added, and then it is diluted with water and made up to mark, shaken well for uniform concentration. Then the absorbance of each solution is measured against blank. So we'll choose a 570 nanometer, around the red color is developed. Then a calibration curve is plotted by taking the concentration of fluoride ion along y-axis and the absorbance along x-axis. According to the principle, as the fluoride concentration increases, the color developed by the complex decreases. It forms a colorless. In the beginning, at 0 without fluoride, the absorbance is very high. As the fluoride ion concentration increases, absorbance decreases. Then, we'll detect the fluoride content present in the given sample water. Fluoride content in water. So, before using the water should be free from the ions. So, already discussed in the principal part. See here, it is necessary to separate the fluoride from the sample by distillation, which otherwise leads to error due to interfering ions. So that's why we have to take around 400 ml first in the beginning, 400 ml of distilled water. To that, 200 ml of sulfuric acid is added. Then distill it. It is continuously heated. Okay, 400 first. In the first, we have to take 400 ml of water, distilled water, and 200 ml of sulfuric acid. Then it is continuously heated. When the temperature of the content is raised to above, if it is the temperature of the solution of the distillation in the distillation flask, it reaches to 180 degrees Celsius, stop heating and discard the distillate. First, distillate should be discarded because it eliminates the fluoride present in the sulfuric acid. Okay, then we have to take it should be cooled to about 120 degree Celsius. Okay, then it is cooled to 120 degree Celsius. Then you have to introduce 300 ml of water sample. Okay, then again the same distilled before. When the temperature reaches to 180 degrees Celsius, stop heating. Then we should take the distillation. Second distillation should be taken. The second distillation contains fluoride ion. The first distillations, the fluoride ion is the process, or it contains the fluoride ion which present in the sulfuric acid, whereas the second distillate that contains the fluoride ions which present in the sample water. Then pipette out the known volume of distillate into 50 ml standard flask. To that, the same way, how to introduce 10 ml zirconyl Spanx reagent and made up to mark. Then measure the absorbance of the solution against blank at the particular wavelength 570 nanometer. So we already got a curve of this type. Okay. So then here the absorbance of this fluoride content is present in the water sample, say it may be some X value, and corresponding to this we'll get the volume, not concentrations, the volume of fluoride present in the sample. In this way we can able to find the fluoride contamination present in the sample water, okay, by colorimetric method.